So let's put spermatogenesis into the larger context. So we have in the male reproductive system, we have testes in males. And inside those testes are the seminiferous tubules. And the tubule is just that. It is a hollow tube. So if we were to look at a cross section of one of those seminiferous tubules, you'll notice that the inside is hollow. It's called the lumen. And you'll see lumens in many different areas. So lumen just means a hollow area. And that lumen is surrounded by tissue. So it's a tube just like a PVC pipe is a tube. You have an outside and you have a hollow inside. If we blow that up a little bit more, you can see here and also in figure 7.2, you can see that we have the tubule wall. And it is along those tubule walls that we have these spermatogonia. Those are the things that undergo mitosis. You have to continually have a set of cells that are undergoing mitosis in order to keep enough of a reserve of that cell that they could eventually specialize into those primary spermatocytes. So remember, we start meiosis with a diploid cell. Primary spermatocytes are diploid. They are produced from spermatogonia. In conjunction with these primary spermatocytes, there's also another cell involved, and it's a helper cell. These primary spermatocytes are not going to undergo meiosis until they get the right message, and that message is a hormone. It is actually these cells that are in accompaniment with these that are called Sertoli cells. They are actually what receives the hormonal message that tells this primary spermatocyte, now it is time to undergo meiosis. So some of you may have had A&P or taken an endocrinology class, and if you remember fish stimulating hormone, FSH, that is the hormone that will tell these to undergo meiosis. So the primary spermatocytes will undergo meiosis 1, producing those two haploid cells. Those are your secondary spermatocytes. They will now complete meiosis 2, and they will create our spermatids. Our spermatids will then mature into sperm. And those sperm are maturing in that hollow space. And then from there, they can begin to swim out of the testes through the seminiferous tubules, through the epididymis, then the vas deferens, and finally are released through the urethra.